Welcome back to Vaccine Design course. Now, under functional analysis of antigenic protein structure to predict the epitopes of antigenic protein, let's look the tool that predict epitope candidates based upon the processing of peptides in the cell. So for that, you need to go to the IEDB website. Under T cells, you'll be getting T cell epitopes processing prediction. So let's look at that. So the second one, which is T cell epitopes processing prediction, click on the first link, which is named as proteasomal cleavage or TAP transporter MHC class 1 combined predictor. So what this tool does is it combines predictors of proteasomal processing, TAP transport and MHC binding. So our interest is MHC binding, right? To produce an overall score for each peptide's intrinsic potential of being a T cell epitope. Okay, so basically it's a MHC1 processing prediction. It's MHC type 1. Here, as you can see, you need to copy the FASTA sequence and paste it here, and then the format you have is FASTA format. You can make it to that, and prediction method, let it be default, which is IEDB recommended. The source species is again human. That's what we need. Now the alleles, let's go for 2 alleles, and length, let's make it all length. Okay, so if you want frequently occurring alleles, you can select that, or you don't have to select it and manually select the alleles. So I'm gonna select the frequently occurring alleles because that's what makes sense. And then I'm selecting some of the alleles of all length. Okay, not specifying any length. Okay, now select the proteasome type, immuno, and maximum precursor extension. So now to understand in detail, go to the help button, scroll down and see what the proteasomal cleavage prediction type is, TAP prediction parameters are, and other results. Okay, now after doing all these or setting all the parameters, click submit button. Now you can see the results page. Here you can download your result, and the first was shown in the result as input sequence, which we have done it. And the next, the tabular one, is you have the alleles, different alleles that you have selected. It's start position, end position, peptidic length. Since we have selected all length, it has displayed all the length type, the peptide, then different scores, proteome, TAP score, MHC score is all written. Now it's the high score, it means it's high efficiency. Okay, so when you click the total score, when you click onto it, it will go from the minimal score. When you click again, it starts with a high score. So here, as the result which was displayed first, it means the first one is the highest predicted one, then followed by the others. Okay, because the total score is 0.97. It combines all the different scores, and again the IC50 values are also mentioned here. Okay, now for again the best interpretation, if you need any help, you can scroll down and look the interpretation prediction results, how it is interpreted. This is the output table, the processing, and the total. So as you can see, it combines the proteasomal cleavage, TAP transport, and MHC binding. So it predicts a quantity that is proportional to the amount of peptide present by the MHC molecules on the cell surface. So if it's a high score, which means it's of high efficiency. Okay, now going back to the link, let's look at the second link. Okay, which is neural network based prediction of proteasomal cleavage sites and T cell epitopes. You have 3 prediction methods in it: NetChop, NetCL, and NetCL pan. Okay, so the commonly used one is NetCL. You can try out the other 2 methods by following the same steps that we are gonna do it now. Okay, again you need to enter the protein sequence in FASTA format. Then what you need to do is see the other parameters, which is the again method specific options, which is by default 0.15 as weight on C-terminal cleavage, weight on tap transport efficiency is 0.05. Then it says about weight is super types and threshold. So the threshold which is set by the software is 0.75. To know about the things in detail, in help it says about the 3 prediction methods, okay, what it says and everything. Now NetChop, it's a predictor of proteasomal processing. NetCL, why I said NetCL means that is a predictor of T cell epitopes along a protein sequence. So that's why we are going to select the NetCell. It also employs a neural network architecture. Whereas NetCL PAN, it's an update to the original one, that is the original NetCL server, which allows the prediction of CTL epitope with restriction to any MHC molecules of known protein. Okay. Now making the others default, we are clicking the submit button. Before that, this page is again T cell epitope processing prediction. It's a proteasome cleavage prediction page. Okay, proteasomal cleavage prediction. Now when you click the submit button, and you will be getting the result. Again, you can save these pages by clicking save all pages. By saving, you will get the tabular result. Okay, so let's look at the table view now, where you can see the prediction score, then peptide sequence, predicted MHC binding affinity, and other parameters. Now about the prediction scores and peptide sequence, both these are the important ones to consider. Okay. Yeah. Now, so the first one means that has the highest efficiency. The prediction score, as you can see, it's increased. Let me just click the prediction score again. Okay, you need to click. Okay, if you click, you will see the green ones which are displayed on the top. Okay, it was not ordered at first. You need to click on the prediction score for it. So as you can see, 1 2 3 4 5 6 7 8 9, there are 9 peptidic sequence, okay, which has the T cell epitope processing prediction, okay, which has the proteasome cleavage prediction. Now so when you look at the chart view, you can see the positive predictions which are seen in green color and negative predictions in the pink color, right? Now you again can go to the help button to see what each parameter says. Okay. So that's about it. We now know how the table view looks like and how the chart view also looks like, and you know that you can save this page by clicking this. If you want to save only a particular page, you can click this button. If you want to save all pages, you can click the save all button. Okay, now try the other 2 methods by yourself and see how it comes. Okay, now if you go back to the main site again, you have the third link, which is MHC-NP, that is the prediction of peptides naturally processed by the MHC. Okay, the second one, it's a predictor. We know that we did the NetCL, which is the predictor of T cell epitopes along a protein sequence. The third one is MHC-NP, which employs data obtained from MHC elution experiments in order to assess the probability that a given peptide is naturally processed and binds to a given MHC molecule. Okay, that's what we are gonna look now. So take the MHC-NP site, which is the prediction of peptides that are naturally processed by MHC. Again, for that we need to paste the protein sequence in FASTA format. The prediction method is MHC-NP. Now what you need to do is you need to select the MHC alleles. So when you see there are 6 alleles, you can manually select them, okay, by clicking all lengths. So it's the third one that I'm selecting. Now it's the 4th, 5th, and the 6th one. Okay, so I'm gonna sort the peptides by percentile rank because I need to know the percentile rank and its positions and its how much it is higher, it has higher efficiency. Okay, so after doing all the procedures, you need to click the submit button. So let's wait for the result. Now at the same time, let's go to the fourth link under T cell epitopes processing prediction, which is MHC2NP, which utilizes the MHC2 ligand elution data to predict the naturally processed MHC2 ligands. Okay, so for that, again, you need to copy your sequence in FASTA format and click the submit button and wait for the results. So here the advantage is we don't need to select the alleles. Okay, it's already the MHC2 one. You have the sequence name, the peptidic start position, end position, length, the peptide, the N motif, C motif, cleavage probability score, and the cleavage probability percentile rank. So go to the help button where it says the step-by-step procedure of what you need to do. Now, as you can see, the result page displays the top 5 peptides, right, per each protein. 1, 2, 3, 4, 5. It's the top 5 peptides per each protein. So each row in the table corresponds to one prediction. It contains the sequence name and other information, right? It's N and C terminus considered for calculating the cleavage probability score and everything. Now, the higher the numerical value of cleavage probability score and lower the numerical value of the percentile score, it's the better the chance of peptide being a ligand. So the first one is better being the chance of ligand. So here it's actually the top 5 peptides per each protein and the first peptide. That is, it starts from G, P, I, S till T. That is, that peptide has a higher chance of being a ligand. That's how the result is interpreted. Okay. Now, when you go to the third link, let's just try the third link again. Third link and select, instead of 6, just select some of the alleles and see if it is working. Okay. Again, you can go to the help and see how to interpret the results. It's pretty straightforward, but then let's do it again. Okay, let's consider 2 alleles, all lengths, sort the peptides by percentile rank and submit it. Okay, so when you do that, then you will get the results faster. So I'll just show you a demo of it. These were the input sequences, and then you have the probability score. Okay, so it's higher the score, more likely to be naturally processed. Okay, so yeah, so 0.7, that's the highest score, which means it's more likely to be naturally processed, which is already mentioned it here. Okay, so this is how you need to interpret the MHC. That is the third one, which is prediction of peptides naturally processed. So these are the peptides which are naturally processed, whereas you know how to interpret the fourth result, right, using MHC2 ligand. So that's all about the T cell epitopes processing prediction, where you have 4 different types of rules. Okay, now let me just show you the step-by-step procedure that we did. First we went to the first link, right? Okay, then the second link we started, and then it's pretty straightforward. You just have to do according to the software it says and then click the submit button. Okay, so that's about it. Thank you.